and thank you for the invite. I was asked by Dr. Salters and Dr. Elaine to present an introduction to schistosomiasis. So that's what I'll do today. Hopefully that'll help you with your project. So I'll first provide a brief introduction to platyhelminthes, the flatworms, and then I'll introduce the digeneids, which is what the schistosoma are. It's a group of schistosoma is involved in. So let me go ahead and start. The platyhelminthes are flatworms. There's about 25,000 species that are known. And as you could see here, they're dorsal ventrally flattened, and they have a dorsal and a ventral sucker. And so these are the general traits of flatworms. They tend to be flat, acoelomates. They lack or have incomplete digestive tracts. They use protonephridia for excretion. They tend to be monoecious, which means that they have both female and male gonads in the same worm. So they're hermaphrodites, with the exception of schistosomes. Schistosomes are the exception to a lot of these traits, but I'll get to that in a minute. And they have attachment devices to attach to the tissues of their hosts. So the schistosomes are within Digenea. About 6,000 species, and only about 12 of those species infect humans. And this is the general life cycle of digeneids. So they have sexual reproduction in a vertebrate host. So this is human schistosomes. That's the male and the female schistosome there. The adult worms will release eggs in the vertebrate definitive host, and those eggs will then eventually enter the environment, usually through fecal or urine contamination into freshwater bodies. When those eggs hit a freshwater body, a free-living stage called a miracidium will hatch, and those miracidia have cilia, as shown here. They swim through the water in search for a mollusk. Usually it's a snail. And then the worms will reproduce asexually in the mollusk and release a second free living stage called a cercaria. Here are different types of cercaria. This forked tail one is the schistosome. And the cercaria will swim through the water, and they will, in some cases, create a metacercaria. In other cases, depending on the life cycle of the digeneid, infect the vertebrate definitive host and eventually form the adult sexual stage. So that's the general life cycle of all digenea. So let's talk about the Strigiiformes. That's the group with the schistosomes. All the Strigiiformes have forked tail cercaria, as shown here. The adults parasitize all classes of vertebrates and many invertebrates, and their location within the host can be quite variable. There are two types of schistosomes that have public health implications. I'll talk about the least important of the two, which is the one that causes swimmer's itch. You may not have heard of swimmer's itch, but it is a public health problem. It is associated with schistosomes that use birds as definitive hosts. And when those cercariae swim through the water and they encounter the skin of a human, sometimes they try to infect the human host instead of the correct avian host. And when those cercariae penetrate the skin of the humans, they can cause pustules and an allergic reaction that can be quite itchy. You can treat it with hydrocortisone and antihistamine creams. But it's a real big problem along a lot of lakes, especially the Great Lakes and other lakes, particularly in the northern part of the U.S. These are this is the diversity of the schistosomes. The ones in red here are the ones that cause swimmer's itch, and the ones in the box here are the ones that cause human schistosomiasis that I'll get to. So this provides you with a sense on the definitive hosts. So there's a variety of definitive hosts of different schistosomes, the different cercariae, eggs, and adults. So you can see they have quite a diversity of morphology in the different stages. So this is the general life cycle associated with avian schistosomes that cause swimmer's itch. So the vertebrate definitive host is usually some sort of waterfowl. And then the eggs are released from the waterfowl, the Myricidia hatch. They reproduce asexually in a freshwater snail. The cercariae is released, and if the cercariae penetrates the skin of the human host, it can cause swimmer's itch. This is a map of some of the states where swimmer's itch occurs. You can see it's quite widespread. And it causes huge economic losses for owners of resorts on many lakes where it occurs. But the real public health issue, the one that causes most of damage across the globe to human health, are the human schistosomes. So these are different electron microscopy images of human schistosomes. So this is the male here and the female here. That's in the gynecophoral canal of the male schistosome. So there's a rich history in this disease. It goes back as far as Egyptian mummies. So schistosome eggs have been found circulating antigens in Egyptian mummies. So it's been around for quite a long time. It was described originally by Theodore Bilharz. So often you'll hear it referred to as bilharzia. So bilharzia and schistosomiasis are synonyms. And it was also described on Egyptian papyri back in the 1500 years ago. It was described as causing pink or red urine, which is typically a symptom associated with schistosoma hematobium. It also had a major impact throughout history. In the 1800s, Napoleon's army in Egypt suffered severely from hematuria, which is blood in the urine caused by schistosoma hematobium. And in World War II, schistosomiasis was brought to international attention. Many of the Allied soldiers were affected in China and the Philippines. At one time, over 3,000 American soldiers suffered from the disease, and it cost the American Armed Services over $3 million during the war. So I'm just going to show a brief video that gives a little bit more information and introduces schistosomiasis. Hopefully this will play just fine. Let me pull it up here on the screen. Just bear with me for one moment. Let's hope that this works. I might need to change the sharing application. Let me change the window here. All right, can you all see that movie? I will start playing it in a moment. I just want to make sure you can see it.
Yes, we can see it. Thank you.
Perfect, great. Well, here, it should play just fine then.
Another bloodthirsty parasite that burrows through our skin is the schistosome. The schistosome is endemic in parts of Asia and Africa and utilizes a snail as an intermediate host before infecting humans. Juvenile schistosomes, called cercaria, emerge from their snail host into fresh water, where they swim frantically looking for the only host where they can complete their development, a human. Once the swimming cercaria find human skin, they eject their tail and utilize skin-dissolving enzymes to burrow through the skin of the foot and eventually into the bloodstream.
One of its remarkable abilities is its capacity to live in what should be the most inhospitable place on Earth, an individual's bloodstream. These worms actually live in blood vessels. Now think of it. If you were an infectious agent, where's the last place you would want to set up shop? Where you're being bombarded daily by antibodies, being bombarded daily by white blood cells, and yet here the schistosome not only survives, but it thrives.
Once the juveniles have matured, the male and females pair up for life, where they mate, continuously producing and releasing eggs.
Let's take the example of the bladder schistosome, known as Schistosoma hematobium. With this type of schistosomiasis, the egg has the ability to traverse through the bladder wall. As the egg does this, it elicits a host inflammatory response that's also associated with bleeding. As a result, people who harbor bladder schistosomes develop blood in their urine, a condition known as hematuria. A curious instance where the presence of hematuria caused by bladder schistosomes affected history was when Napoleon entered Egypt. Napoleon's troops, it was prophesied, would be stricken by Pharaoh's curse should they choose to enter into Egypt. And what was Pharaoh's curse? It was male menstruation. And sure enough, when many of his troops came into Egypt, they acquired bladder schistosomiasis, and many of the troops believed that this was the prophecy of Pharaoh's curse fulfilled.
Another fascinating parasitic disease.
All right, so we will return to the PowerPoint now. I like to try to intersperse some videos so you're not listening to me the whole time. Let me go ahead and do that. All right, hopefully you're able to see the PowerPoint now. Should be the case. So another interesting aspect about schistosomiasis is that it ranks second among all parasitic diseases in the world in how debilitating and devastating it is to humans. So it ranks second behind malaria. So major parasitic disease worldwide, with 200 to 300 million people infected. Many are school-age children, and I'll tell you why that's the case in a minute. With about 20 million suffer severe consequences and about 800,000 deaths per year. In 1993, it ranked as the main disease burden in children between the ages of 5 and 14. And the disease is actually increasing. There is a drug to treat the disease called praziquantel, but when humans return to freshwater bodies where the snails and the parasite are prevalent, they get reinfected very quickly. So this is schistosomiasis prevalence by age. You can see it peaks around the age of 10 to 13 years old. It's believed that adults acquire immunity to these infections, which is why you see a decline in prevalence with older age classes. So as I mentioned at the start of the lecture, schistosomes that cause human schistosomiasis have very different traits than the majority of digeneans and flatworms. First of all, they're not flat. They're actually round, as you can see from the photos here. Their eggs do not have opercula. Opercula are little flaps that allow the worm to escape the egg. So that's also something different from most other flatworms. They only have one intermediate host, which is the snail, and one definitive host, which is a human, whereas many trematodes have more than one intermediate host. They are dioecious, meaning that they only have, they're also sexually dimorphic, meaning that the gonads, female gonads, are found in the female worms only, and the male gonads are found in the male worms, and the males and the females are morphologically different. The female worm is smaller than a male worm in most cases and sits in the gynecophoral canal. The male is larger and has larger suckers because the male attaches to the veins in the human host and the female just lays in the gynecophoral canal. The other thing that's pretty impressive about these worms is that they exhibit monogamy. When a male and a female find one another in the host, they mate for life. It's believed to be the lowest taxonomic level to exhibit monogamy. And so those are some of the traits of the schistosomes that are a bit different than a lot of other flatworms. So you might be wondering, well, why are they not flat? It's believed that the round shape evolved secondarily because they infect veins, and the veins are cylindrical. And so it's a lot easier to attach to the inside of a vein if you're flat than if you are round. And so it's believed to be a secondarily evolved morphology as a function of the environment in which they inhabit. So there are three major species of schistosomes that infect humans. There's Schistosoma japonicum, and it is found in the anterior mesenteric veins that drain the small intestine. There's Schistosoma mansoni. It's found in the posterior mesenteric veins that drain the large intestine. So there's a schistosome that is found in the veins in the small intestine and in the large intestine. And then finally, Schistosoma hematobium, which causes urogenital schistosomiasis. It's found in the veins that drain into the bladder. And so that's why it causes bloody urine, because it damages the bladder, which causes urine or blood in the urine. So this is the general life cycle of human schistosomes. So they reproduce sexually in the human host. Then those eggs that are released from the female will enter the environment through either feces for Schistosoma mansoni and japonicum, or urine for Schistosoma hematobium. Those eggs, once they hit fresh water, will hatch, and the miracidia will be released. These are ciliated, free-living stage that swims through the water and is looking to infect one of three genera of snail hosts. There's a separate genera for each species of schistosome. They reproduce asexually in the snail, and they release a second free-living stage called a cercaria. And the cercaria has these forked tail, and that's how it moves through the water body. And it's swimming through the water body and is attracted to chemicals released by human skin. And if it's successful enough to penetrate the human skin, which is what is shown here, then it will migrate through the bloodstream to the heart, the lungs, and then the liver, and then eventually make its way into either the intestines or the bladder, depending on the species, where if it's lucky enough to find a member of the opposite sex, it'll reproduce sexually for life in a monogamous state in the veins in those locations. We're going to skip the video describing the life cycle. If there's time at the end, I'll go ahead and do that. But I think we've probably covered enough detail on the life cycle thus far. So these are the three genera of snails that harbor each of the species of schistosomes. So Biomphalaria is the genera that will harbor Schistosoma mansoni. Balinus is the genus that harbors Schistosoma hematobium. And Oncomelania is the genus that harbors Schistosoma japonicum. And this is an infected Biomphalaria snail from my lab releasing cercaria. You can see all these little things swimming through the water are the hundreds, if not thousands, of cercariae being released by the snail. So each one of these is capable of infecting a human. So they can produce massive numbers of these parasites. And this is the distribution, geographic distribution of the various species. So Schistosoma japonicum is found predominantly in Southeast Asia and areas like China, Japan, and other parts of Southeast Asia. Hematobium is exclusively found in Africa. And then mansoni is found predominantly in Africa, but it was unfortunately brought to Latin America, mostly northwestern and western Latin America with the slave trade, and it's now endemic in Latin America as well as parts of the Caribbean. All right, I think you've heard enough of me for a bit, so let's take a break and watch a video here that describes schistosomiasis in greater detail. Give me one moment as I pull that up. All right, hopefully you can see that movie. Can someone just verify that we're good?
Yes, we can see it.
We're good.
Great, I'll start it. It's just about 2 minutes.
On Animal Planet's Monsters Inside Me, John Figge was infected by a schistosome parasite that swam into his brain and reproduced into a mass of eggs and worms so big that doctors thought it was a tumor. Fortunately, those doctors were able to save him. But while John Figge was able to find relief from these parasitic worms, millions of other people around the world aren't so lucky.
It's estimated about 10% of the Earth's population are infected with this, and that's a lot of folks.
Dr. Fred Lewis's lab is ground zero for research on the parasitic worms that cause schistosomiasis. They're funded by the NIH to provide samples of the parasite to researchers around the globe.
We collected these today from primate number of mice, and people will use these for various vaccine antigens to test their vaccines.
To provide those samples, they raise the parasites through the multiple stages of their lives. They start by infecting snails just as the parasites do in the wild.
These are infected with the parasite. The parasite will mature in the snail at this temperature in about 4 weeks. No, I kind of know what I'm up against. After all these years, I get used to it.
After the parasites leave the snail, they look like this. They're desperate for human flesh to burrow into so they can mate and start laying eggs. To show us just how desperate they are for human flesh, Dr. Lewis presses his finger to the bottom of a petri dish. The fatty acids from his skin are enough to trick the parasites into thinking they've found a human host.
They will think that they have found intact skin and will try to penetrate into it.
At this point, the lab uses mice rather than humans to allow the parasites to mature into egg-laying adults. Researchers are using these samples to learn everything they can about these parasites and how they develop.
What we're trying to do in this lab is to identify what genes make some snails resistant.
Controlling this parasite worldwide remains a huge challenge, including the search for a vaccine, because
the parasite has evolved mechanisms where it thwarts immunity. So we're trying to find ways around that.
Because the parasite needs unsanitary water to spread, it's mostly a problem for people who live and visit impoverished tropical regions, just as John Figge found out in the show.
All right. Let's head back here to the PowerPoint. Lost my page. Oh, there we go. All right. So you can all see the PowerPoint, correct?
Yes, we can see it.
Wonderful. So as it was mentioned in the video, the brain infections are rare. So I just wanted to walk you through some of the symptoms and issues that schistosoma worms cause in humans. And so one of the big questions researchers were asking for quite some time is, how do the eggs get out of the body? The females produce large amounts of eggs. We know that they get out in the urine and feces. And what was discovered was that most of the eggs are forced through the human body tissues by the inflammatory response initiated by those eggs, which is associated with the immune system. So the host immune system is what forces those eggs through the tissues and then eventually into the fecal material or the urine. And so many of these eggs actually don't ever get out of the body. So about 1/3 make it out of the body, and about 2/3 are actually successfully attacked by the immune system and surrounded by granulomas. So you could see here, this is an egg, and this is what's referred to as a granuloma. These are a series of motile immune cells, mostly eosinophils, macrophages, and neutrophils, that surround the egg, release toxic materials to that egg, like hydrogen peroxide, and then they wall off this egg in the tissue of the human host. And so that's the inflammatory response and the granulomas that are produced. Like I said, 2/3 don't make it out, so they end up in the lodged in the tissues of the human host, usually the liver, spleen, and bladder. So liver and spleen and intestines for Mancini and Japonicum, and a lot of them end up in the tissue of the bladder for hematobium. And so over time, as you build up these granulomas, they cause scar tissue. The release of the hydrogen peroxide and other compounds by these immune cells causes damage in and around the egg, and that creates scar tissue, fibrotic tissue, and over time, as that scar tissue builds up, you get liver damage, spleen damage, intestinal damage, as well as bladder damage, and then those tissues don't function the same way they did before those infections. So most of the pathology associated, excuse me, I'm getting a call. Most of the pathology is associated with the inflammatory response to the antigenic nature on the eggshell. The adults actually don't cause any pathology at all. As you heard in the video, they're under the radar of the immune system. They actually coat themselves with lipoproteins of the host, so they're not detected by the immune system. So there's a few stages to the infection. There's the migratory stage. This is when the cercariae penetrate the human host. It's often asymptomatic, but can cause a dermal rash like swimmer's itch. Then there's the acute phase. This is associated with egg production by the worms. You can get headaches, chills, fever, gastrointestinal discomfort. For Schistosoma japonicum, it's often referred to as Katayama fever. And then you start getting the buildup of granulomas surrounded by the eggs. But the chronic phase, this is really a chronic infection. The chronic phase is really what causes the disease. It's the buildup of these granulomas and scar tissue over time. For Japonicum and Mancini, you can get abdominal pains, fibrosis, scar tissue. 8% of the infections develop liver fibrosis. You can get enlarged spleen and liver. It can stunt the growth of children, causing dwarfism. You can occasionally get eggs that lodge into the brain and cause neurological damage, although it's not really super common. And you could even get cancers that develop. The long-term impact is the loss of tissue function. You also get ascites with Japonicum and Mancini infections. This is accumulation of fluid in the peritoneal cavity, loss of liver function that causes high blood pressure, and then in the portal veins, which then causes fluid to diffuse out of those blood vessels into the peritoneal cavity. So this is a fluid buildup as well as enlargement of the spleen and liver, which is causing the symptoms you see here. The symptoms for hematobium are a bit different. You rarely get ascites or enlargement of the spleen and liver because the infections are associated with the bladder. You will get blood in the urine. You'll get bladder damage, loss of bladder and kidney function, pain associated with urination. You'll also get urogenital issues, so issues associated with the vaginal region for women. And bladder cancer is actually somewhat common in chronic infections. It's so common in some areas that children actually believe that their urine is supposed to be pink. So there's some really cool special adaptations of schistosomes, such as antigenic camouflage. Like I said, the adults and larvae are under the radar of the immune system because they coat themselves with lipoproteins of the host. The cercariae use chemoattraction to find snail hosts and human hosts. And the other really cool adaptation of the worms is that the cercariae are released only during the day, usually at the peak heat of day, when humans are most often in water bodies. So let's finish the lecture with some discussion of epidemiology. So the transmission of these worms is closely associated with human use of water bodies, shown here. So in places like Africa, Southeast Asia, and Latin America, where there isn't running water, many humans will have to go to water access points to get water, and that's typically where they get infected. It's also an emerging disease. It's increasing in prevalence associated with irrigation as well as dam construction. So you all have a project associated with the Senegal River Basin where I work. And it turns out that when the Diama Dam was constructed there, that was associated with massive increases in prevalence of schistosomiasis in the region. This is not uncommon. The same thing occurred with the Aswan Dam construction in Egypt before the Diama Dam was constructed. So this is a common phenomenon. You get dams that are created, you get more irrigation canals, you get greater contact with water body, more habitat for snails, and you get an increase in schistosomiasis. And I know that all of you are going to be developing control programs. I don't want to give you too much information on control. I just wanted to hit on some high-level bullets. Some things that are being used to reduce schisto are education. So many people are not aware of the life cycle, so educating humans on the life cycle can have an impact on transmission. As I mentioned early in the lecture, there is chemotherapy. Praziquantel is a drug that will successfully eliminate schisto. When humans return to freshwater bodies, they will get reinfected. There is host control, so controlling snails. And there's a lot of researchers working to develop a vaccine. However, there is no currently effective vaccine for human schistosomiasis. So with that, I think I've gone about 35 to 40 minutes and wanted to make sure that there was some time for questions. So hopefully that is a nice overview, an introduction to schistosomes that cause public health problems. And I'm going to turn it over to Dr. Salters and others at this point in case we have time for questions.
Thank you. Thank you so much, Dr. Rohr. That was, for me, I knew a little bit about schistosomiasis, but that was pretty eye-opening. There was a lot about them that I didn't know. So thank you very much for that. We will open it up to questions at this time. If those of you on Zoom, if you have a question, please put it in the chat. Here in the room, I'm looking out at people. Be thinking about your questions. I want to just ask a question about Senegal and when the dam was built there. Since there was that prior experience in Egypt, do you know of, was there any sense of the need to prepare for the idea that schistosomiasis would, the rates would go up, or what was historically, what was the thinking on that?
Not that I know of. I don't think there was a whole lot of learning that occurred, but I could be wrong. You know, there is MDA campaigns, mass drug administration campaigns, yes, throughout Senegal. So there is distribution of praziquantel. So in the region that I work in, when we provide praziquantel to the villages that we work in, and then we return a year later. So we're giving praziquantel to school children. When we return a year later, prevalence in most of the villages for schistosoma hematobium is back up to 70 to 90%. So in a year, most of those kids are getting reinfected, and 70 to 90% of those kids are infected. So it's pretty extremely high levels. It's a major hotspot globally. Mancini is usually around somewhere between 20 and 40% from year to year. So it's lower levels for Mancini in that region. So even though they're using MDA, prevalence is still quite high after the treatments.
Okay, thank you. Thank you for that. There's a question in the chat.
Which population groups would you say are most receptive to information about the disease when you're doing outreach to share about the disease?
So we've had really a great reception when we provide educational material to the villages that we work in. So we worked in 16 villages in our clinical trial and 23 villages in our broader work. They were all very interested in our work. They were interested in learning in ways that they can reduce schistosomiasis. We are beginning a study in 88 villages in the region, and we will be providing them. So that'll be a study where there are 4 treatment groups. We will be providing a fourth of the villages education on the public and private benefits of our intervention. Another fourth will get education on just the public. Another fourth will get education on just the private. And then the last quarter will receive no education at all. And so, so far, we're excited about that work. I won't tell you too much about the intervention because I think the paper was provided to the class, and I don't want to say too much about control efforts because I know that that is a major thrust of the project. But we've had really great reception, and the people that we work with are thrilled to have us working in the villages. And I could even show some photos if there's time, but when we tell them what we're doing, oftentimes we'll get many villagers just volunteering to help us with the work that we're doing. So they'll jump right in the water bodies, they'll help us with our experiments. And so that's been really great because we're hoping that we will get a great adoption of our innovation in the future.
Continue on here. Okay. How is schistosomiasis diagnosed? Is there any biomarkers?
Yes, great question.
Is there an instant testing kit available?
So the way it's done in Senegal and across most of Africa is we get urine samples or fecal samples, and they are looked at under the microscope for the presence of eggs. There are some, qPCR tests. I don't know of a really reliable instant testing kit. We probably should be working in that direction because ELISA tests should probably work. But the sort of gold standard is CATOCATS, which is a filtration, fecal filtration and microscopy approach, and then looking at urine samples directly under a microscope.
Are you connecting with local governments, nonprofits, or religious organizations?
Yes. So we have working relationships with the health organizations in Senegal, so the ministries of health. We have to actually get approval through the ministries of health and coordinate with the Ministry of Health in our clinical trial work in Senegal. So the Senegalese Ministry of Health is aware of our work. We're in the process now of jumping through those same hoops to getting approval to do work in Kenya. So yes, we're working with governments, we're working with local governments. We also have relationships established with local universities in the Saint Louis Richard Toll region in Senegal. So we work with faculty members at the University of Gaston Berger in Saint Louis, Senegal. And then we also do all of our clinical trials and our ecological work in Senegal with the assistance of nonprofits there. So they receive subawards from our grants as well as faculty members at the universities. And they end up implementing a lot of our work when we are not there. We try to be there a few months of the year, but it's a challenge to be there all 12 months of the year, especially for me, given that I have teaching responsibilities and I'm the chair of the department. And so those nonprofits are really important as well to make sure that our research is maintained when we are not able to be in those countries in Africa.
Thank you.
You're welcome.
Hi, so I wanted to make sure that this was clarified, but you were talking about how about 1/3 of the species will actually leave the human body and 2/3 will inhabit in habitat within the body. In the life cycle, do they have a way to naturally die? Like, do they have a way to, like, do they, in our blood, you said that they adapt to those white blood cells and to the things that most species would absolutely hate being, and they thrive in that environment. So what does kill them? Like, does anything kill them naturally, or can our body do that?
Yeah, great question. So the adults will die natural deaths. So so if you let them remain in the human host without treatment, they will die of natural deaths, and the glycoproteins on the surface of the adult worms that prevent them from being detected by the immune system break down, and then they get expelled by the immune system. So the adults are under the radar of the immune system. The eggs they release are not, and that's what triggers the inflammatory response. And the white blood cells then will kill the eggs and wall off the eggs. But in the process of doing that, you get a buildup of scar tissue and fibrotic tissue. And only about a third of those eggs are pushed through the bladder wall or through the intestinal walls to successfully get out of the host. So over time, all those eggs and scar tissue accumulate, and that's what causes the damage. But there is natural death, but most of the adults are not killed by the immune system. They end up either dying from praziquantel exposures or natural deaths in the human host.
Hi, I also have a question. You said that like with building dams and irrigation systems, it usually increases the prevalence of like infection. Is that because like you're altering the water so that it's like more still water, or just that like higher contact with water itself is what like makes it increase?
That's a great question. I don't know that we know all the different mechanisms. So we are currently doing a study where we are looking at schistosomiasis levels throughout Africa before and after dams are constructed. And we're also looking at different types of dams because there are hydroelectric dams. There are also irrigation agricultural-based dams. And then there's dams of varying sizes, from really small dams to really large dams. And then, so what we're doing is we're trying to quantify how the irrigation canals in the regions before and after dams have changed. We're trying to figure out whether agricultural dams are associated with greater increases in schisto than hydroelectric dams. But we think there's several mechanisms. So we think that there's increased water contact associated with the reservoir behind the dam. So that provides a lot more perimeter for humans to contact water, because the reservoir is a larger surface area than the original river was. And then the reservoirs are usually constructed to either for irrigation, for agricultural dams to pump water for irrigation purposes. So you get a lot more irrigation canals, which means that there's a lot more water contact. For hydroelectric dams, it's usually a reservoir to control the flow of water, so that way they can modify electricity production. But the other thing that we've shown is that fertilizer and some pesticides can actually increase snail production and schistosomiasis risk. And so it may be more than just increasing the amount of water and habitat for the snails. It may be that the agricultural dams also increase agriculture in the area and fertilizer use. That promotes the snail habitat and food. So snails eat predominantly attached algae, and they live in predominantly submerged aquatic vegetation. And algae and vegetation tend to thrive when fertilizer runs off into the water, because they need nutrients. And so we're seeing evidence that fertilizer use can also promote schistosomiasis. So there may be multiple mechanisms by which dams are increasing schisto.
Thank you.
You're welcome.
Other questions? Are there any more in the chat or? Okay.
How much does the drug cost that does kill the disease, and is it readily available? Who produces it? What's the control of the drug?
So it's cost pennies to make a pill, but when you incorporate the delivery expenses, it depends on how you do your calculations. It's probably somewhere between 10 cents to $2. And like I said, it depends on how you deliver it. So if you just distribute it through schools and ask school teachers to deliver the drugs, which is how it's often done. It can be extremely inexpensive, on the order of 10 to 20 cents. For more remote villages, it can be more costly to get the drugs there, but it's usually 10 cents to $2, so incredibly cheap. And that's why many organizations like the Bill and Melinda Gates Foundation are supporting purchasing praziquantel and distributing it throughout Africa and other parts of the world. The challenge is that it's not sustainable. So as soon as those kids or adults return to those water bodies, they're getting reinfected. And so what the World Health Organization is promoting now is the combination of mass drug administration with the control of intermediate hosts. And so they're hoping that the combination of those two approaches will be more effective than just giving drugs. The other concern with drugs is that there is evidence of resistance development in the worms. So the more drugs that you deliver, the higher the likelihood the worms will evolve resistance, and then those drugs become futile. They become useless because the worms are resistant to the praziquantel. So very, very inexpensive for the most part to deliver these drugs to people in Africa.
Thank you. We have one more here.
Hi, can you just maybe explain a little bit more how the medication like actually works in the body and like its mechanisms a little bit, if you know?
Yeah, I don't know a whole lot of details about the sort of cellular and mechanistic pathways, but it targets the adult worms, and it doesn't last very long in the body, and that's why the reinfections happens once these individuals who are treated return to the water bodies. I probably should know more about the mechanisms, but because I'm predominantly an ecologist, I don't delve into the mechanistic details on the praziquantel and how it ends up killing the worms. So good question, and I probably I know the answer, but I don't know it off the top of my head. It does not impact the larval worms, to the best of my understanding. And so if there are larvae that are migrating in the body, they tend not to be impacted and can establish soon after the drugs wear off in the system.
Thank you. Did you have a follow-up? Oh, okay. Okay, thank you. Other questions here in the room? I think we may have hit everything everybody can think about.